Well, hello, AP Bio students. Mrs. Shelton here. I am going to talk to you today about our Unit 2. So this is we're on review now. You should have done Unit 1. Hopefully you watched the screencast and you looked at kind of the outline that College Board gave us as well. Hopefully you guys also did the Unit 1 practice FRQ that I gave you and just timed yourself and getting prepared for what the timing of your FRQs are going to look like as well. For Unit 2, we are talking about cell structure and function. Now with this was back at chapter 4 and 5, so it's been a while, but fortunately, this is a lot of information you've already had through your regular biology class, plus in your middle school as well. But again, we're taking it to that college level. So there are some more concepts that we do need to make sure that we are reviewing and going over. So let's go ahead and kind of start. We're going to use the course exam and description and just what these enduring understandings and some of the learning objectives that College Board specifically sets out for us in order to make sure that we are understanding what we should be going through. Okay. So first of all, their first topic, 2.1, so cell structure and subcellular components. Now, again, with this, I have provided you just a cutout of what's on the CED, but you can go through the presentation and then pull up each slide and look more specifically at them. I'm not going to read them word for word for you because, oh my gosh, you guys can read. But in essence, what we're actually trying to just understand here is that we just need to make sure that we are recognizing the structure and function of a lot of subcellular components and organelles. So including, you can look at just some of the words that are here, we understand what ribosomes are. We need to make sure we're understanding what the rough versus the smooth ER, but actually here's an exclusion statement that actually says we don't really need to worry about the specific functions of the smooth ER. We need to make sure we're understanding the Golgi complex, and then we also need to make sure that we're understanding your mitochondria, lysosomes, vacuoles, and chloroplasts. So when we are actually talking about structure and function of subcellular parts, we definitely need to know, first of all, just similarities and differences between prokaryote cells and then plant cells and animal cells of eukaryotes. We need to make sure that we understand that ribosomes, so ribosomes is ribosomal RNA, absolutely imperative for making proteins. You can't make proteins without ribosomes. So all cells have to have ribosomal RNA subunits. So this is also evidence of the unity of all life as well. Now, when we start talking though about eukaryotes, this is where we start getting our compartmentalization and understanding that for protein synthesis, the role of the rough ER is going to have ribosomes embedded in there. Remember, smooth ER, that's going to be more for detoxification. Your Golgi complex is going to be for modifying proteins for export. Remember, your mitochondria is for aerobic respiration. Lysosomes are going to be your packets. So membrane packets filled with hydrolytic enzymes. Vacuoles are going to be for storage. We'll talk a lot about the plant vacuole quite a bit. And then remember, your chloroplast is going to be your organelle that is for photosynthesis. But make sure you're going back through and you are recognizing, again, just those differences, what they look like, and also the function of those. Some illustrative examples that College Board does specify in here. So this term here, we haven't heard this particular term. So glycosylation. So glycosylation and other chemical modifications of proteins. Really what they're asking you to understand is this role of this endomembranous system. This is going to be an important part. We did address the endomembranous system as far as how we manufacture proteins and this role of the ribosomes that are going to be creating that first primary structure of a protein, but then also through this rough ER, we have our transport vesicles and then also the Golgi apparatus that's going to be modifying these proteins. So making sure the shape is proper, but then also adding just other chemical modifications to these proteins in order to create their function. Now, when we get to our cell membrane structure, then you'll also see proteins that do have carbohydrates attached to them or their lipids or protein lipid combinations as well. And that's all through these modifications of making proteins through this endomembranous system. So really don't freak out when you hear a word like this that could potentially get used. Just make sure you're recognizing the role and the connectivity of the rough ER and also the Golgi apparatus in manufacturing and modifying proteins for their ultimate function. Now, in 2.2, so here, cell structure and function. So some of the learning objectives, this is understanding how components of organelles contribute to the function of the cell. So here they're talking again about the endoplasmic reticulum, the mitochondria, lysosomes, vacuoles. They also want you to understand some of those structural features and those structural features that allow for organisms to capture and store energy, which really what they're talking about here is make sure that you know that structure and how the structure of the mitochondria and the chloroplast really play a role in aerobic respiration and photosynthesis that we'll get to in Unit 3. But in this one, we need to make sure that we're understanding those structures a little bit better. Now, one of the concepts they need you to understand is making sure that you understand how different cells have different functions based on these components. So what I did for you is I just want to talk to you about different cells that have high concentrations of endoplasmic reticulum or different cells in our body that might have high concentrations of mitochondria and just what that's going to do and how that's going to change. So here, if I have a cell, like here in my pancreas, the beta cells of the pancreas, they produce insulin. Insulin's a protein. So I would expect to have structures that are a lot of structures that are designed for making massive amounts of a protein. So I'm going to expect to see high amounts of rough ER in those beta cells of my pancreas. Now, we know that muscle cells require a lot of energy. So here I've got a picture of cardiac muscle fibers, and these cardiac muscle fibers are going to have high amounts of mitochondria in order to generate high amounts of ATP. Now, macrophages. Macrophages are a part of your immune system that are actually going to be taking out just old cells. They're going to be engulfing bacteria or foreign particles that come in. Well, these are going to be cells that have high quantities of lysosomes in there. Remember, lysosomes are going to be packets of digestive enzymes that are going to fuse and break down these particles. And then when we talk about cells that are going to have high amounts of vacuoles, well, remember the vacuoles in plant cells actually play a big role. It's not just water storage, but they also, remember here, that these vacuoles are going to increase the amount of water, decrease the amount of water. They're going to be ion storage, sugar storage, all sorts of things, but they also then play a role in this turgor pressure as well as not just storage of materials. So kind of keep in mind why we would see some of these different organelles in different concentrations in different specialized cells. Now, I told you that we are going to need to talk about the mitochondria versus the chloroplast and making sure we understand, first of all, similarities between mitochondria and chloroplast. Remember that your mitochondria and chloroplast both are double membrane structures and both of them have circular DNA. This is evidence of that endosymbiotic theory. So with that, we'll talk a little bit more about that later, but we need to recognize that these are both for energy transfer. And so these are excellent, excellent organelles for energy transfer. And so when we talk about the mitochondria, we know that this is our site of aerobic respiration. And with this double membrane structure, we've got our outer membrane, our inner membrane. Remember, our inner membrane has lots of folds, increases that surface area, and this high concentration area of protons. So we're going to talk more in Unit 3. We'll talk more about our chemiosmosis and electron transport. But our electron transport happens in this inner membrane. This intermembranous space is where our high proton concentration is going to be built up. And then we've got our mitochondrial matrix in the middle. And so that matrix in the middle is where our protons are going to be diffusing back through. And this is also where we'll talk more about our Krebs cycle and making sure we recognize that the Krebs cycle is going to be taking place in this mitochondrial matrix. Now, in our chloroplast, remember again, double membrane structure, but those two membranes don't serve a specific purpose other than provide us evidence of that endosymbiosis. Now we need to recognize our thylakoids. Remember, our thylakoids are these membrane sacs. This is where my light reaction is going to be taking place. This is also where chemiosmosis is going to be taking place, electron transport. And then remember my Calvin cycle, where carbon fixation is going to occur, and now I'm going to be making my G3P molecules. That's going to be happening out here in the stroma. So do go back through and review your basic structures of your chloroplast and also of your mitochondria. And that's just going to help you when we now move on to Unit 3 and talk more specifically about the processes of aerobic respiration and of photosynthesis. All right, this was the part of the unit you guys loved, which when we talked about cell size. So if we take a look here on what is necessary to understand, this is our surface area to volume ratios, making sure we understand that cells stay small. And why cells are small is because the surface area to volume ratio affects its functionality. Remember that the surface area, or the plasma membrane, that's regulating what can come in and out, the exchange of nutrients from outside the cell to inside the cell. Remember, we calculated surface area to volume ratios. There's all sorts of different ways you can calculate it. You will have an equation sheet if they give you a cube or a square or a rectangle. But what you need to recognize is that volume always increases at a cubed value, where surface area is at a squared value. So as cells get bigger, volume is going to increase at a much faster rate than surface area does, and that's going to limit how big a cell can be. Remember, the more surface area it has compared to volume, the more efficient it is at diffusing materials and moving materials in and out of the cell. So keep that in mind. Now with this, when we talk about these surface area to volume ratios, you got to be able to calculate it, but then you will also have to make sure that you have practical application as to how these specialized structures, how these different strategies in different cells have actually maximized movement in and out. So here's a diagram you're all familiar with. So we did a lab where we dealt with cubes and we just measured different boxes of different shapes in order to calculate surface area and volume. So here's just a simple example, just showing you a cube that's a 1 by 1 by 1, and my surface area to volume ratio is, so here's my total surface area is 1, and then here is my total volume, which is, or my surface area is 6, because I have 6 sides that I had to add up. My volume then is 1, so this would be like 1 centimeter cubed. This would be 6 centimeters squared. So remember those values. My surface area to volume ratio would be 6. Now, if I increase this box size by 5 times, so now I've got a 5 by 5 by 5 box. Well, now all 6 sides added together, because I take my area of one side, multiply that by all 6 sides, and now it's 150. My volume, which is now 5 times 5 times 5, 125, my surface area to volume ratio now is a 1.2. So I have definitely decreased my surface area to volume ratio greatly in this process. So now if I had that same volume, but now instead of having just one giant cell, I now have all these individual cells filling this space, well, now I've got a tremendous amount of surface area, and I've now maintained my surface area to volume ratio. So cells are much more efficient by staying small, having more cells, and so each cell then can be highly efficient and effective at transporting materials in and out. Now, when we do talk about some different illustrative examples that we should know. So here is what College Board would want you to be exposed to, and just understanding some of these illustrative examples of surface area to volume ratio. So this is something we didn't get a chance to talk about since we left for COVID-19, but in root cells will actually have these little extensions coming out of the root cell that are called root hairs. And so these cells, because remember, and this is where the plant needs to be obtaining water from the surrounding environment. So by having this process that extends out, well, now I've increased surface area to allow for more water movement into the cell. So root hairs, so these root hair cells would increase surface area. So that's an illustrative example. Now in your intestines, so when it says gut epithelial cells, well, here are my intestines are for digestion and absorption of nutrients. So here in my small intestine, if I need to increase the surface area, well, each cell is actually going to have not only villi, these extra folds of the cell membrane, but then I'm going to also have these little microvilli as well on top of them. If I look at these cells here, I have a tremendous amount of surface area because I've now folded that cell membrane so many different times. Very similar to the inner membrane of the mitochondria. There's way more surface area in the inner membrane than outer membrane of the mitochondria because of those increased folds. Now, guard cells, we talked about the stomata, and we talked about the stomata being those pores that are going to open and close in order to allow for carbon dioxide to come in and oxygen to escape. Also is where water escapes from cells, from plant cells as well. But what controls those stomata are these guard cells. Now, these guard cells actually have a tremendous amount of surface area on them, because with that too, they're able to increase their turgor pressure by increasing water moving into the vacuoles. And so that's going to actually make them swell back up. And that's going to open up this stomata, this opening. Oh, remember my lovely example of my stomata. But guard cells are, again, another example of cells that have a very high surface area compared to volume, but this allows them to adjust the amount of water moving in and out in order to then be able to open and close at different times of the day. Now, another example that College Board would like for you guys to be aware of is just in general, how does surface area and volume relate to how other organisms on a large scale interact with the environment? And here I just pulled out a couple of examples here of this change in surface area. So if I look here at a jackrabbit versus an Arctic hare, different environments here. Well, this jackrabbit has bigger ears. And again, this increased surface area of the ears is not to hear better, it's actually for heat exchange with the environment. And so this is a way for excess heat to dissipate from this jackrabbit, where now this Arctic hare doesn't want to lose body heat, so it has smaller ears. Same thing if we look at our two different elephant species, where I've got my African elephant that has very large ears. Those large ears are not for hearing, those large ears are for exchanging heat with the surrounding environment to get rid of excess heat, versus my Asian elephant that doesn't need to do that as much. And so they've evolved to have smaller ears. So our fourth topic, plasma membrane. So with the plasma membrane, we need to make sure that we understand the basic structure of the plasma membrane, and we also are making sure that we understand our fluid mosaic model of the cell membrane. So here's a diagram that should look familiar to you. So remember our lipid bilayer, our plasma membrane, our phospholipid structure. Remember your phospholipids are amphipathic. So where your phosphate heads, these are polar, and we would call these hydrophilic. The tails then are fatty acid chains, and so those being nonpolar, those are hydrophobic. So we end up forming this lipid bilayer, and that lipid bilayer then ends up giving us a semi-permeability to molecules that can actually pass through them, depending on their size, and also depending then on their polarity and their charge. Now, because this is going to limit what molecules can move in and out of the cell, we also need to have lots of proteins embedded into the plasma membrane. So most of these proteins, so remember I've got peripheral proteins and then I've got my integral proteins. And so most of these integral proteins, these are going to be channels in order to allow for transport of charged particles, or also for active transport. We'll talk about different transport mechanisms. We've got some glycoproteins with carbohydrates attached to the proteins for cell-to-cell communication. And again, this would be that example of a protein modified. So this protein got this carbohydrate attached to it by the Golgi apparatus. That would be that illustrative example I went over with you too. We also have to make sure that we're remembering cholesterol, because this cholesterol is embedded in here. Remember, cholesterol is a steroid, this being a lipid. So this is nonpolar, and it can embed in this middle layer, this hydrophobic layer. Well, cholesterol is going to maintain fluidity of the cell membrane. So remember that in animal cells, remember that these fatty acid chains are most often saturated fats, where in plants, they will often create more unsaturated fatty acids, so that's going to maintain fluidity of the membrane. But for animal cells, they need this cholesterol embedded in there to maintain fluidity, because through fluidity, we can increase permeability of molecules in and out. Which brings us to membrane permeability. So with this, what are the structures that influence that selectively permeable membrane of the cell membrane? And then also the role of the cell wall. So when we do talk about the lipid bilayer, you need to absolutely remember just that lipid bilayer alone, what it allows to pass through are very, very small hydrophobic molecules like oxygen, carbon dioxide. These would be molecules that readily diffuse right through. Something like water or some uncharged polar molecules. These can get through, they're limited, but that's why we'll talk about like something like aquaporins, but those small uncharged particles. Large uncharged polar molecules, however, like glucose and sucrose, those cannot get through unless there's a facilitated diffusion that's happening, and then also ions. So if it's a charged particles cannot get through that lipid bilayer. So we need to make sure that we understand oxygen, carbon dioxide, those can readily diffuse. When I talk about my polar molecules, most of my polar molecules cannot pass through unless it's very small, like water. Water just simply bombards it. And then remember my my non-polar molecules can actually pass right through, and we'll talk about that when it comes to some permeability of things like some steroid hormones. Charged particles, though, those can't get through, so they need help. Now remember the cell wall. Cell walls are outside of the plasma membrane. All cells have to have a plasma membrane. So don't just say, oh, plant cells have cell walls. That's such a big mistake that ends up happening. Yes, plants have a plasma membrane, and then they have a cell wall for structure, and it also actually plays a role in what can come in and out of the plant cells as well. But then also remember that fungus, so all of your fungi are going to have plasma membrane and then cell wall, and then you're also going to have some prokaryotes. There are some prokaryotes that also have cell walls as well. So just keep that in mind. Now for concept or topic 2.6 on membrane transport. So here they need you guys to be able to describe the mechanisms that organisms use to maintain solute and water balance. So what's our passive transport mechanisms and our active transport mechanisms? And then also what are some of these mechanisms for moving materials, these large materials across the membrane? So this is where we need to make sure that we are addressing exocytosis and endocytosis in order to understand these basics. So here is very simple diagrams that you guys have already seen, our passive transport mechanisms that are going to be moving molecules from high concentrations towards low concentrations. These do not require energy. So we'd be talking here about diffusion. We'd be talking about facilitated diffusion, where now I need to have this doggy door type of protein, so this channel protein, or I also have these carrier proteins that also would be our facilitated diffusion examples. But these are all passive transport, no energy required movement of molecules from higher concentrations to lower concentrations. Now remember that active transport requires energy. So here I see this input of ATP, and active transport, we're moving against the concentration gradient. So we are moving molecules from low concentration towards a high concentration, and that requires energy in order to do this. If I need bulk transport for large particles or for large quantities of ions or large quantities of molecules, whatever it is, I can use exocytosis. Well, I can't, but my cells can use exocytosis or endocytosis. And this is that engulfing or using the plasma membrane in order to create these vesicles, these transport vesicles for export in exocytosis, or for import, which would be my endocytosis. But it's using that cell membrane to engulf and transport bulk material. Now here's some other examples of transport that you should be familiar with. We talked about co-transport. With co-transport here, this is a diagram that we have where the cell is actually using proton pumps, creating this high concentration gradient. And with that high concentration gradient, then having something like a sucrose being a larger molecule, now, sucrose, I can move into the cell when I allow this diffusion of protons to also move from this high concentration to this low concentration too. So co-transport would be something to make sure that you are familiar with or you're remembering. And then also another example here of a pump, an active transport that's absolutely essential when it comes to functioning of our nerves and sending action potentials. Here I've got my sodium potassium pump. And with my sodium potassium pump, here's my input of ATP, but I'm establishing a concentration gradient of sodium on the outside of the cell, potassium inside the cell. Remember, on my neurons, I set up a salty banana, but the sodium potassium pump sets up a resting potential for cells. So then when I need cells to send our neurons to send a signal, well, then I've got voltage gated channels that can open up in order to allow for this diffusion of sodium in and potassium out in order to send action potentials. But that's another example of different transport mechanisms. Now, concept 2.7, we just need to make sure that we're recognizing our facilitated diffusion. And so with facilitated diffusion, remember that this is a form of passive transport. So here's just a few more examples just of facilitated diffusion. We use the term aquaporins. So remember that water, some's going to get through, some's not going to get through. So actually when I need water to be able to osmose a lot, well, now I can actually just set up these, well, the cell can actually have these protein channels that are specific for water called aquaporins to allow water to simply diffuse through. Now, I can also have, like what I just described with action potentials, I can have action potentials where I've got voltage gated channels that when voltage changes across the membrane, then maybe my sodium channel opens up and sodium can then diffuse through. But through my other ion channels, I do have ligand-gated ion channels. This is actually what happens at the synapse of neurons. So neurotransmitters are ligands. These are molecules that will trigger the opening of these ion channels and ions rush through, which now triggers an action potential in another neuron. There's also some ion channels that are always open in order to allow for diffusion. And then I have some that are mechanically gated as well, where now through pressure, they can open or close. But ion channels would be examples of, with this, there's no active transport happening here. So there's no ATP. They're just receiving different signals of, oh, do I open to allow for that diffusion to occur or not? So just recognize some special types of facilitated diffusion that can happen. All right. Here's where you look and you see what is written on this page for tonicity and osmoregulation, and you start freaking out. Remember, you don't need to freak out. You see these little symbols, these size for water potential, and go, oh no, that looks so scary. But remember that water potential, what we're talking about is how concentration affects water movement through cells. Water potential is just that measurement. We already know that with diffusion, molecules move from high concentrations to low concentrations. All we need to remember with water potential is water will always move from high water potentials to lower water potentials. Remember that water potential is partially due to pressure potential and solute potential. So how much dissolved solute do I have in that water solution? And then also how much pressure is pushing against the membrane as well is going to contribute overall to that water potential. Here we have to relive the terms hypo, hyper, and isotonic solutions. And then we also have to be able to recognize this equation. If we ever have to calculate our solute potential, this equation would be given to you, all this information would be here, and if you're ever asked, it's a simple plug and chug type of equation in order to calculate your solute potential. So here are the pictures that you guys have seen before, animal cells versus plant cells in hypertonic, isotonic, and hypotonic solutions. So these are terms that are describing the outside solution. Remember, these are frames of reference, but in this particular case, we are talking about hypertonic solutions. Hyper means more, so there's more dissolved solute in the solution compared to the cell. So The cells themselves are actually hypotonic compared to the solution. And so remember, I also told you too, water will always move from the hypo towards the hyper. So in this case, water is moving towards that more dissolved solute. So these cells are going to shrivel. Here in a plant cell, if I over-fertilize my lawn, well, now I've created a hypertonic solution surrounding my plant cells. And so water is going to leave the cells towards the hypertonic dissolved solute. My cell itself was hypo compared to the hyper on the outside. Water moves from hypo to hyper. And so in this case, my plasma membrane has pulled away from my cell wall. And so this is where plasmolysis occurs. Here's isotonic. In isotonic, this is what animals always try to maintain their internal environment in isotonic solutions. Because in isotonic solutions, my tonicity, my dissolved solute inside the cell and outside the cell then is the same. Now, in a plant, however, that's not exactly ideal for a plant because now I'm not having increased water coming into this vacuole and increasing the turgor pressure. So isotonic solutions often leave plant cells flaccid, so they don't have the same internal strength and internal pressure that they need for that support. Now, hypotonic solutions. In a hypotonic solution, remember water always moves from hypo to hyper. So if you injected yourself with pure water, well, you're going to cause your red blood cells to lyse open because water is going to move from this hypotonic solution towards the dissolved solutes that are inside the cells. And so then that cell membrane isn't strong enough to withstand that pressure and is going to burst open or lyse. Now, in a plant cell, water is going to move into the cell, but now what happens is as vacuole fills up, this actually creates an internal turgor pressure against the cell wall. And so here, the plant cell is not going to burst open because the cell wall is actually protecting it from now overfilling. So recognize that plant cells prefer to be surrounded by hypotonic solutions. Animal cells prefer to be surrounded by isotonic solutions. And understand that flow of water and why that's going to occur. Now, here is something that will, again, cause stress. You're seeing a YouTube problem, but I selected this picture just to help you remember water potential. If I have pure water over here, pure water in an open container is going to have a water potential of 0. Because remember, if it's an open container, the pressure potential is 0. There's no solute, so the solute potential is also 0. So over here, I have a water potential that's 0. Over here, it's an open container, but now I have dissolved solute. Remember that in calculating your water potential, that solute potential is actually going to be a negative number. And that's oftentimes where, if you're calculating water potential, students get confused because they forget that water potential with a negative, this is a smaller number than a 0. And so water is going to always move from higher water potential to a lower water potential. And so that's the basics that we need to make sure that we're understanding is that flow of water is always from high to low. Remember in biology and AP Bio, when in doubt, everything moves downhill from high to low. Now, if I do take a look at my plant, and water potential and how it leads to the movement of water through a plant, if I take a look at where my water is going from the soil to the trunk of the tree, well, I'm going from higher water potential to lower water potential. Well, where's my absolute lowest of lowest water potential is out here in the outside air that's a negative 100 megapascals. So again, remember negative 100, this is a really, really low number compared to my higher number of a negative 0.3. So remember to be thinking with water potential in negative terms. And remember also my units for water potential, they could be megapascals or they can also be bars. Doesn't matter. Pay attention to the numbers. And if you're asked to write down that water potential, never leave a naked number. Use the units that they always give you. But this is why water moves from the ground up through the plant and then out that stomata is because one, because of adhesion and cohesion, but also just that water potential. There's going to be higher to lower water potentials as we move up this tree and then out into the air. So keep that in mind. Water always moves from higher water potential to lower water potential. Here's some other illustrative examples of the importance of moving of water. So here I have this little amoeba here. Amoebas have a contractile vacuole. So contractile vacuoles, now if I've got this water living protist, well, it's got to be able to regulate because it's got dissolved solute in here. So if it's in fresh water, well, water would just be flowing in and causing this poor guy to burst open. He's not going to be able to survive. So by having this contractile vacuole, the contractile vacuole can actually be drawing water in and then actively pushing that water out in order to maintain proper tonicity and maintain an isotonic environment with its, so inside the cell versus outside the cell. Now, you do not need to know all of these transport mechanisms that are in a plant vacuole, but I just wanted to show you this picture so you can recognize that plant cell vacuoles are pretty complex, actually referred to as a tonoplast, and there's lots of different active and passive transport channels that can actually change the tonicity inside the vacuole to either be able to draw more water in or more water out. So be able to just kind of recognize why vacuoles in plant cells are so important. They're not only for storage of ions and for proteins and for sugars, but they also maintain that turgor pressure for the plant cells that's also so important for structure. Now, in concept 9, 2.9, mechanisms of transport, I really just don't see how that's any different than things we've already talked about. Describe the processes that allow ions and other molecules to move across membranes. We already addressed that. So moving on. Concept 10, 2.10 is understanding compartmentalization. So we talk about cells and the fact that we have all of these different compartments for doing different jobs. And so when we talk about organelles in eukaryote cells, we do reference them as being membrane-bound structures. Now do remember that ribosomes are not membrane-bound structures. These are subunits that are going to be performing part of protein synthesis as far as translation is concerned, but we've got to remember that when we talk about membrane-bound structures like the endoplasmic reticulum, like the Golgi apparatus, like the mitochondria, like the chloroplast, like our lysosomes especially, we need lysosomes to stay in their membranes because one, that allows for specialization there, but it also makes sure these digestive enzymes are concentrated, and these digestive enzymes aren't going to be now breaking down other molecules where we don't need them to be. Also remembering that with this compartmentalization, now I can actually have, with increased compartmentalization, different locations, I can actually have within here, I can have specialization within that cell. Some things we have to remember too when we do talk about mitochondria and chloroplasts is remember that with chemiosmosis, I'm creating high concentration gradients of protons. Well, increased protons means higher acidity or dropping of the pH, which now that could denature proteins somewhere else. So it's absolutely vital that a lot of these reactions take place in these membrane-bound structures. So we're not essentially affecting other proteins in other areas in the cell. The last concept that we need to make sure that we are addressing, we already addressed it, and I talked about the chloroplast, we need to make sure we understand these origins of cell compartmentalization. How do we actually have these specialized locations? The biggest part that I need you guys to make sure that you are remembering here is that you are remembering the endosymbiotic theory. How did we get mitochondria and chloroplasts? Because when we actually look at some bacteria that are aerobic bacteria, they actually use their plasma membrane for electron transport and their surrounding environment. Then they've got ATP synthase that's also embedded in there as well. And so they are actually performing aerobic respiration, but they are not a eukaryote. But it's also a single membrane structure. Well, now ancient anaerobic prokaryotes, if they engulf them through endocytosis, well, now I've got a double membrane structure. And now I've got this symbiotic relationship with this used to be ancient prokaryote and this eukaryote that are now working together, providing a nutrient source, providing it um, a habitat in order to live. Now, our evidence of this endosymbiotic theory is the fact, again, that both of these structures, our chloroplast and our mitochondria, double membrane structures, and both still have circular DNA. Circular DNA is only in prokaryotes. Eukaryotes have linear DNA. So that's another evidence of this endosymbiotic theory. So this is where, here we've got our photosynthetic prokaryotes. We're engulfed. And then this is where we have our plants, and we do have plant-like protists that actually have mitochondria and chloroplasts together. And then we also have our animals, our fungus, that have mitochondria only. So we need to kind of make sure that we're remembering that. Again, common mistake you guys make is you just assume that plants have chloroplasts only and don't have mitochondria. Remember, plants don't need us. We need plants. That's the basics. So with that, that is a lot of Unit 2. Unit 2, it was, even though it was only 2 chapters, it was 2 really big chapters. So do make sure that you are going back. Most importantly, make sure that you are referencing different transport mechanisms would be what I would want you guys to make sure that you are focusing most on. So with that,